Hi, my name is Ananya Garg. I'm a rising senior and a biology major. This summer, I studied the activation of potential EGR1 targets in lens cells after cataract surgery. The reason I'm focusing on cataracts is because they lead to one-third of blindness worldwide. Cataracts are the ocular clouding of the lens, and they are solved through cataract surgery, which has side effects including posterior capsular opacification, or PCO. This is also known as a secondary cataract, which occurs when remaining lens epithelial cells cause scarring. It is known that scarring is caused by transforming growth factor beta, but we want to know more about the mechanisms behind how this process occurs. To learn more, my lab conducted RNA sequencing specifically at 6 hours to see what was happening at this early time point. We found that 3 major pathways were upregulated. The first pathway is the positive regulation of inflammatory response. This can help us get a better understanding of the potential role of inflammation in PCO pathogenesis. The second pathway is the positive regulation of smooth muscle cell proliferation. Myofibroblasts are similar to smooth muscle cells and are major contributors to PCO. The final pathway is the positive regulation of epithelial cell migration. During the early stages of PCO, epithelial cells migrate to the posterior side of the lens where they will proliferate. We also found that early growth response 1, also known as EGR1, is upregulated at this time. Interestingly, EGR1 is known to upregulate various profibrotic markers. The staining shows it to be robustly activated by 6 hours post-cataract surgery in my lab's mass cataract surgery model. Now that I know EGR1 is present, I wanted to see what other genes it could have the potential to regulate through bioinformatic analysis. This analysis allowed me to find several interesting potential EGR1 targets with known EGR1 binding sites in their promoters. 2 interesting upregulated targets are SERPINE1 and thrombospondin1. SERPINE1 is an inflammatory mediator, and thrombospondin1 is a fibrotic marker that participates in wound healing response. From this data, I can conclude that the lens epithelial cells are profoundly affected by 6 hours post-cataract surgery. There is a large upregulation of genes involved in inflammation, proliferation, and migration, which are all known responses of the eye after cataract surgery. A notable gene that is upregulated is EGR1, which has the potential to upregulate a number of genes which could be important to the inflammatory response. My future work includes immunofluorescence staining of these targets in our post-cataract surgery model mice. And to ensure this upregulation is dependent on EGR1, I would also stain for these targets in EGR1 null mice. I could also perform QRT-PCR to further validate this data. I would like to thank the Duncan Lab and Dr. Duncan, Adam Ferranda, and Samuel Novo for being mentors to me throughout this project. Thank you to the National Eye Institute grant and Summer Scholars for funding.